Hello, everyone, and welcome to Talk Nerdy. Today is Monday, February 27th, 2023, and I'm the host of the show, Cara Santa Maria. And like always, before we dive into this week's episode, I do want to thank those of you who make Talk Nerdy possible each week. Sometimes you'll hear me read ads. Sometimes you'll hear these programmatic pre-baked in ads that I'm not reading. Those actually pay at a much lower rate. But the bulk of the way that I can keep the show afloat is from support from people just like you. All you've got to do is go to patreon.com/talknerdy, and you can read about the different tiers. You can read about how to get an ad-free episode. And yeah, it's just so lovely that you guys year after year continue to make sure this show is possible so that everybody can listen to it free. And, you know, only those who can afford to support it are doing it. You're really kind of offsetting it for those who can't afford to pay. So this week, I do want to thank the top patrons. They include Daniel Lang, David J.E. Smith, Mary Neva, Brian Holden, David Compton, Gabrielle F. Jaramillo Gonzalez, Joe Wilkinson, Pasquale Gelati, and Ulrika Hagman. Thank you all so much. All right. So before we get into this week's episode, I do want to let you guys know that it's looking like I'm not going to have a new episode next week. So we're going to be off the week of March 6th, and we'll be picking back up the week of March 13th. And that's because I am leaving, as of my recording this, not as of it airing, but as of my recording this, I am leaving in a few days to head to the Middle East, to head to Jordan with an organization at the school, the clinic, the psychology services center where I am doing my internship. So although I go to school in California, I'm finishing my PhD in California. Each person who gets a PhD in clinical psychology, and PsyD, by the way, is required to do a 1-year accredited internship. And it's often not at the place where you go to school. So my internship is at Nova Southeastern University in Fort Lauderdale, Florida. And I found out after I started here from a trainee that I work with that they have an incredible organization called the Humanitarian Global Organization that travels each year to places where there is a need for psychological services. And so there's a chance that I'll be going to Southern Africa later in the year. But for now, we're headed to Jordan, and we're going to be traveling all over the country. In particular, we're going to be focusing on a domestic violence shelter for women and children. We're going to be focusing on doing some trainings in a local university there. And then I think it looks like we're going to be spending a little bit more time in one of the largest, or actually it might be the largest Syrian refugee camp in the world, and doing some assessment and intervention there. So I'm going to be away for a little while focusing on that. I'm going to be out of pocket, and I just wasn't able to scramble between all of my coursework, writing my dissertation, podcasting, doing all the things that I do too many things. I wasn't able to scramble and get enough episodes in the can. So I think we're going to be skipping next week, and then we'll come right back out for the following week. So March 30th, we'll be back on air. Okay. That's enough preamble, because this episode is so much fun. We had an absolute blast. I'm going to have her back on the show. I have to, because she's fascinating, and her book is so exciting. So basically, I had the opportunity to chat with science and technology writer Sally Adee. She was the features editor at New Scientist. She has written for so many different outlets and appeared, let's see, where has she written? She's written for the New York Times, The Economist, BBC Future, Quartz. She's been on multiple podcasts, she's been on Radio Lab and The Economist Intelligence Podcast, BBC Breakfast. And she was also the science consultant for a television adaptation of Naomi Alderman's The Power, which is on Amazon. She's won multiple awards, and I love this. She is a citizen of Germany and the US. She currently lives in London, so when we were chatting, it was kind of the middle of the night. So keep that in mind too. She's brilliant and fascinating, and she wrote her very first book. It's called We Are Electric: Inside the 200-Year Hunt for Our Body's Bioelectric Code and What the Future Holds. So without any further ado, here she is, Sally Adee. Well, Sally, thank you so much for joining me today.
Thank you. Thanks so much for having me. This is very exciting.
Yeah, so I'm excited to talk about your new book, We Are Electric: Inside the 200-Year Hunt for Our Body's Bioelectric Code and What the Future Holds. And I've got so many questions right here at the top, because you are a science writer, and you've written for many outlets, but specifically you, or maybe I should say most recently, you were a features editor at New Scientist. So you've covered like basically every aspect of science.
Mostly. I mean, I was a bit weak on the sort of Large Hadron Collider kind of stuff, but
Weak, okay.
so particle physics not really my thing. But the sort of neuro tech stuff was sort of my bread and butter, and I really enjoyed writing about history of science stuff and editing those sorts of features, and just basically, I guess, I sort of developed an interest in the bigger picture behind how science became, how the various disciplines became what they were.
Right. And you've worked on like different publications, you've been on different podcasts, you've even been a consultant before on a TV show. Is this your first book?
It is my first book. Yeah. I mean, it took like 10 years to write it because I mean, I don't think I was going to write a book, but then the more, so let me sort of back up a little bit. So I started out my career um, at an engineering magazine in New York. Actually, it's called IEEE Spectrum, and that's the Institute for Electrical and Electronics Engineers. So I was on the semiconductor beat, sort of microchips and the sort of transistors that are in them. And one of the things that made this beat worth being on was that you could start really getting deep into neuromorphic chips, which are the kinds of microchips that are sort of, they're trying to model them on the way that the human brain works, and also on sort of neurointegration, brain computer interfaces and like advanced prosthetics that like link up with the nervous system. And I just thought that was so cool. And, the Defense Department was, they sponsor a lot of this research. They put a lot of money into it. And, for various reasons, good and bad. But they would have these conferences and they would show these advanced prosthetics that were like, they were wild. I mean, they were so amazing, and you would see them and it just felt like that the future was going to be here really quickly, and it was just really, just overwhelmingly cool brain computer interfaces that could sort of, do remote work, just by tapping into your brain signals. Anyway, I got really obsessed with this, and I started learning about all the sort of the nuts and bolts of it, what made it work. These little Utah arrays, these are like little pin cushions that look like a bed of nails for a ladybird, right? You press them into the cortex, and then the 100 little needles pick up your brain signals, the spikes, the action potentials from your brain. But every time, so this all seems to make sense, but it's one of these like stories where you're just like, well, how does it work? Like, how does it work to feed like, electronics into, meat, basically?
Yeah, into our wetware.
Yeah. So what's the interface? Like, how does it work? And, this is where things just got a lot hazier. People had these sort of, I guess, not pat answers, but sort of, ready-made answers. So, for example, there was this whole transcranial direct current stimulation thing, which is the brain zapping that is where they apply a very small amount of current to headgear, basically, and it is meant to make you more focused. So, as with everything, again, I asked, like, well, how does this work? And then I got this answer where it was like, well, the neurons that fire together, wire together, which is the basis of learning and plasticity of the brain. And then if you change the way that neurons fire, then you can make people learn faster. And that's all well and good. But then I was like, well, how are you making the neuron fire? And then it was like, well, we're not sure like which neurons are working. And so I guess this is just sort of a long-winded way of saying I never got like a clear answer that made sense to me. And then I tried to push sort of further and further. And then finally, I got so obsessed that I just decided to quit my job and just research this all the time. I was reading books like, I don't know, they just had these like horrendous titles that were just like, on, gap junctional ion channel currents and like, the glial cells or whatever. I was just so, I was finding myself in the library, like, what the hell am I doing? Like, what am I looking for? And then finally, you find these researchers and they tell you, well, all the stuff that we thought about bioelectricity, that it was just a really straightforward thing about neurons firing. That's only the tip of the iceberg. And while it is correct in some ways, there is so much more going on that people just aren't really reporting on. Or if they were, understandably, if people were reporting on it, it was very it was sort of disconnected. You'd have articles about, bioelectricity's role in regeneration. And it's just like, can we read minds by tapping into the brain's electric signals? And I really wanted to have this I wanted to sort of collect and assemble all the different aspects of this research, and just put it into, the good research. And I just wanted to put it in one place, almost just for myself to have a reference, just so that I could get it in one place, because I hadn't seen it in one place, like for popular science, for somebody like, like me who could to understand.
So wait, how did you make money while you were doing this?
Wow.
If it took that long, I mean, you were still writing, right?
Yeah,
you were still,
no, I was. Okay. So I was freelancing. But also there were, you as a freelancer, you also, there are some jobs that you take that are not sort of public facing, and you just sort of, you write stuff that
you do what you got to do.
Yeah, exactly.
Yep, I do it too.
Yeah, so so various organizations or whatever, and that's actually how I got hooked up with The Power, the Naomi Alderman adaptation for Amazon Studios. So
right. The television consulting you did.
Yeah, exactly. So that was pretty good. So I would read scripts and like apply my newfound pedantry to it and be like, I'm sorry, but what kind of ion channels would do that? They'd be like, oh, please leave us alone.
So you mentioned ion channels. I feel like before we even get too into the weeds, maybe we can take a step back and what is bioelectricity? Like, we are electric? Like, obviously, yes, we are electric. But for people who are like, I don't remember taking neuroscience. I don't remember excitable tissue.
Yeah. Oh, God.
What is the role of electricity within our own cells?
Okay, well, let me take you back to the year 1790. Sure. Sorry. Don't worry. This won't hurt that much, I promise. But yeah, so, right. So 1790-ish, everybody's still trying to sort of come to grips with this sort of general phenomenon of of electricity, sort of writ large. It's still this incredibly mysterious phenomenon. We still don't understand that lightning is the same thing as like the sparks that you get from rubbing amber and stuff. So we're still trying to put it together. And this anatomist called Luigi Galvani, he contends, and with really good replication studies, basically some of the first, he actually contends that there is that it also is active within our body, that it is what is responsible for the nervous system being able to sort of convert your thoughts into actions and your feelings into, your sort of, that you can interact with the world and feel it and move in it at will. And he called this animal electricity. And
this was post-animal magnetism, yeah?
This is sort of all tied up. I think this was
all around the same time.
Pre-animal magnetism, because we still were working out that magnetism and electricity are related. Okay. I'm not sure how far along we were at the time, but
right, because that was Mesmer, but this is Galvani.
Yes, exactly.
So Mesmer Galvanism.
So Mesmer was one of the quacks that kind of came out of the flap that I'm about to discuss, which is where Galvani gets into this huge scientific spat with Alessandro Volta, who is is going to be the inventor of the battery. And Galvani basically says there is a thing called animal electricity, and Volta says there is no such thing. You've just got, this sort of, he had this theory of two metals and the two different substances, whenever they got in contact, would generate like regular electricity. So he objected to the idea that there was a particular kind of electricity at work in the human body. He did not agree that that was possible. And so they have this incredibly consequential war. They're mostly polite to each other, but it's their friends that basically go to town on each other. And this turns into such a massive sort of gyre that every scientist in Europe essentially is forced to choose sides. And some of them swap sides. And, there's all this very sort of Twitter energy going on. But in the end, unfortunately, even though Galvani writes this amazing paper that actually delves into this and shows, it's one of the most elegant papers that's ever been written, I think. And he should have been vindicated, and everybody should have been like, oh, let's immediately begin studying bioelectricity. In the end, it's Volta inventing the battery that sort of changes the terms of the debate almost, where it's like he invents something useful and everybody's like, well, see, Volta was right. And it's like, no, he wasn't right. It's just that he made a thing and you guys are making a category error. But basically, Volta wins the argument. Galvani dies. This is a whole tragedy. Yeah, it's a whole tragedy because Napoleon basically took Volta's side, and there was a whole thing with the papal states being now under, Napoleonic control. This is slightly off topic, but in the end, Galvani was quite devoted to his religious faith as well as science, and he refused to swear an oath to the new republic. And so he got stripped of all his titles, and his wife had died, and he was brokenhearted. He had no more sort of reason to live, and he died. And and even though, so this is very tragic. His nephew, Giovanni Aldini, is actually the guy who probably inspired Mary Shelley to write Frankenstein, because he had been working with Galvani for ages. He was really, really involved. He probably wrote a lot of the ripostes between Galvani and Volta. He probably ghost wrote a few of them. And he was really passionate about making sure that his uncle's legacy didn't slip into just disrepair, right?
Galvani. I was going to ask.
Yeah.
Do you think if not for Aldini that we wouldn't know the name Galvani? A lot of people know because they know galvanism. They know this at least conceptualization. Obviously, we know the name Volta too because of volts.
Yeah, exactly,
that one's pretty obvious. But a lot of people, especially those who study psychology, biology, they've sort of have some fuzzy history of this era.
Yeah.
And we know that name. But is that because of Aldini? Would we not know Galvani if his nephew hadn't been taking up the cause?
Do you know what? Sadly, I think it's the opposite.
Okay, all right.
When I was looking at this, poor Aldini, he started out with these wonderful intentions where he was like, I am going to save my uncle's legacy. But there was nobody of Galvani's sort of stature, he was a real polymath genius, right? And there was nobody like that to take the study of bioelectricity forward. And Aldini tried his best, but he got kind of sucked into the showmanship thing. He used to, he's the guy who reanimated corpses of recently killed prisoners. There's some really grotesque experiments, and those guys were only the tip of the iceberg. He was doing stuff he was not reanimating, but
he was sending massive electric current through their bodies, right?
Yeah, and he was doing, and it's funny, because people know about the stuff that he did with this guy George Forster, this hanged criminal, but he, what they don't know is that he was doing all kinds of stuff. He not only was sort of zapping with, sort of Volta's proto batteries, he was zapping, big animals and small animals, but also there was this weird thing where he had started this galvanic society where what they did is they were trying to make batteries out of biological materials. So this one guy, sorry, I'm going to have to read this to you because
No, it's really cool.
this
This is
is
awesome.
well, this is so weird. So this within this galvanic society, because a lot of people were trying to sort of join him to make to try to sort of advance the study of bioelectricity in this very sort of slightly lesser way than Galvani had done it. This guy, Monsieur Lagrave, is a French guy in the French Galvanic Society. He made a battery out of layers of human brain, muscle, and hat material soaked in brine. So this is like body horror. This is like Saw or something. And they were doing a lot of this. Like there's another scientist called Matteucci. He made a battery out of frog thighs, and he like stacked them. Like he basically like carved them into like the shape of a half orange and just stacked them. And he was able to elicit, like he was able to use the current that came from that to make another frog's like leg twitch, which was the only way we knew at the time whether we were getting a biological current. Anyway, so these kinds of experiments were really tittillating. People were
right. They're like parlor tricks.
Yeah, exactly. So like high society would come and they would just like watch this, and they'd be, they'd watch. In one case, one of the dead guys, he sort of when he stimulated his. Sadly, I believe this was he connected like one wire to an ear and one to the rectum. I may be misremembering this, but the guy actually sort of sat up and his arm raised, and I don't know if this is newspapers of yore sensationalizing the way we do today, but it's apparently he lifted his arm, and it looked like he was pointing at one of the onlookers, and somebody promptly fainted. So this is the kind of thing that Aldini started to sort of veer into, because that he didn't really have a theory, I guess, of bioelectricity the way that his uncle had, and so he couldn't really move it forward. And he succeeded, I think, in blurring the line between like the legitimate study of bioelectricity and the charlatans that were going totally nuts at the time, like Mesmer, like you were talking about Mesmer. He was one of the ones. People were selling like galvanic penis belts and like rings, and just like they were supposed to cure everything from like gout to cancer to Victorian flavor diseases like excessive masturbation, or like like loss of vigor due to
right.
Yeah.
I guess we forget sometimes that, even today, I shouldn't say even today, definitely today, that when it comes to like pseudoscience and charlatanism, some of it is about the show, and the show is how you raise money from wealthy donors. But some of it is about peddling snake oil. It's literally like patent medicine.
Yeah. And I think like there's always such a huge, the placebo effect does so much. You create an expectation in someone's mind, and then it's like, isn't your hysteria cured, lady?
Right.
Or whatever. I mean, there was one thing that I was trying to track down, but I just don't really have the, I'm not conversant enough in the precise language of the time to have figured this out. But there was, I was trying to hunt this down for ages, but there was a thing about in the early 1800s, they were using electricity in one UK hospital to help with like stilted menses or something like that. And you're just like, I think I know what you mean. I think I know what this is, but I can't really. What I'm saying is it was just used for absolutely everything. And of course, we have no records of whether it worked.
I covered on a recent episode of another podcast I work on, the Skeptic's Guide to the Universe. I covered, you've probably seen, you might have even written, you probably wrote about this, but the newish article that was published where a historian was trying to dig into like the historical use of the vibrator. One woman once wrote a book and she was like, the vibrator was used to cure all this. And then they're like, no, there's actually like very few references to that. And it's kind of like, it becomes its own lore.
Yeah,
after a while.
exactly. It gets its own momentum, right? It just, yeah.
Yes.
And
Yes.
it's so easy once you've got this stuff in the cultural water, it's so easy for people to then, you know, just be completely, well, the placebo effect takes over. You're just like, well, this works for
Absolutely.
people, and so it will work for me.
And so then you try it on literally everything. Because the thing with the vibrators, everybody's like,
Yes.
oh, it was used to cure hysteria. It was used to cure hysteria. And it's like, no, they weren't like actually. I mean, there is some documented things where like doctors were literally using it on women's clitorises to give them orgasms to cure their hysteria. But more often it was used all over the body. It was like, your elbow hurts? Put a vibrator on your elbow. Your eyeball hurts? Put an eye, and it's like the radium craze. Like
Yeah. Oh,
just take some
completely.
radium, you'll be fine.
Yeah. Yeah. And electricity was the same. And, you had these
Yeah.
very interesting structures. Yeah. So it was like the list is just as long as, it's such a long list of stuff, gout and, dropsy, epilepsy, everything.
All
And
the fun old stuff like
yeah, I know.
dropsy, wow, consumption, all of it.
Yeah. And, the way that these things were marketed was also very familiar. There was one guy, Perkins, and he made these things. He's watching Volta and Galvani have their fight. And so he's just like, he's in Connecticut and he's just like, I think I can make something out of this. So he makes these things called tractors that are supposed to work on like the bioelectricity of complementary metals or something, whatever it was. It made no sense, but it didn't have to, because like it was sort of within this conversation that was becoming increasingly familiar. He sells these things. He gets actually drummed out of the medical society in his town in Connecticut. They move to England, where he starts giving free review samples to people. It's like the ancientest grift, the most ancient grift. And these judges, I guess, apparently George Washington bought a set of tractors. I actually tracked this down, so this is legit.
Okay.
And but people are like dragging them around their body, and they're like, "This will cure my tumors." But yeah, I mean, it's, snake oil is always such an interesting cultural production. I don't want to, obviously derail or talk too much, but I just find it so interesting how.
No, I love it. Derail away.
Yeah, because like how much buy-in certain things just like pick up cultural cachet at certain times, and then everybody believes it, and like that must contribute to the fact that sometimes these things work. Right. And even though like later you look back and you're like, how the hell did that work? And then, but at the time, I don't know, there's that whole like expectation effect, right? Like
it's huge. Yeah. It's, and like we think of the placebo effect, we think of, it's exactly what you said. It's not that a placebo is actually effective. It's that there are non-specific effects that are at play that are effective. So things like expectation effects, white coat effects, like all of these things.
Yeah,
right?
but like
somebody's
they
paying
create
attention to me.
Yeah.
Like all of that.
Yeah. That one is huge. But also like that does create like beneficial changes sometimes in the brain.
Oh, absolutely.
I think,
but
like
it's
the
not
dopamine
just like,
and all that.
Yeah, there's so many downstream changes. I mean, we think about therapy and how much, but I think it's, I'm always so worried about actually using the word placebo, because by definition, a placebo is an inert substance.
Yes.
Like it's not the placebo that's doing it. It's your relationship with the placebo. It's actually a bunch of non-specific effects that are measurable, tangible effects. We just don't often measure them. You know what
exactly.
I mean?
And like and placebo also always sets it up as if it's like you're being fooled. And it's just like, well, actually, this could be like super powerful medicine if we could harness it. There's that whole, I don't know if they still have it, the Department of Placebo Studies at Harvard. I don't know if there's,
that's fascinating.
Yeah. No, I did a story. I edited a story on this like ages ago at New Scientist. And I was like, oh my gosh, they're trying to sort of harness it. But yeah, like the placebo effect is tough, because if you lean in too hard, people will be like, oh, wait a minute, should I be skeptical? And then it stops working. It's kind of like Tinkerbell or something. Like you have to believe in it.
Right. It doesn't work. It's legit unethical for a physician to prescribe a placebo. Like you can't do that. Like you can't give drugs and tell them they're taking drugs when it's not drugs. Like by definition, that's bad medicine. So it's anyway, that is a full derailment, but I love it. That's what I love about this show. Okay, so where do we go from here? I'm so curious, because obviously we're talking about the history. I think we can spend more time in the history if we want to, but I'm also curious, like what we know now, because there's a big jump from magnet.
this is where
electricity.
this is how I derailed us. We started
I
talking
love
about
it.
ion channels. I'm sorry.
Yeah, no, it's fine. It's fine.
This is very bad. Yeah, this is a really bad habit of mine. This is totally how it happens. Anyway, so I'll take you through just like a tiny speed run from, when we're talking about Galvani and Aldini and all these like electro quacks. So basically, after Aldini sort of screws the pooch, basically there's, yeah.
No, all that.
So basically there's just like this, it's a desert for about 40 years of bioelectricity research, because people are just like, this is just quackery. Like people start cottoning on pretty soon, right? You've got like people being like, all right, this is all BS, basically, right? Like, because it's not that it was BS, it's just that they were sort of throwing shit at the wall and seeing what sticks. And they had no theory of like how zapping people randomly was supposed to work. So they just sort of zapped people. Maybe 2 of them got better. Everybody else just sort of went on being sick. And, pretty soon people were like, look, this is all a bunch of crap. So for 40 years, this stuff is in the desert. And then people, these sort of, greats of science of the 19th century start to get involved. Alexander von Humboldt, Emile Dubois-Reymond, all these guys, these neurophysiologists start sort of putting together the discipline of what we now know today as neuroscience, because they start mapping, the nervous system and figuring out how the action potential works in it. Like, yeah, this is electricity. Then all of a sudden these guys take over again and they're like, look, the study of bioelectricity is legit, but only within the context of the nervous system. Everywhere else, it's crap. So this is that becomes canon for another 100 years or so, because now you've got sort of the legitimate scientists, sort of giving a home to bioelectricity. It comes out of the cold of the quacks.
I have to interject for 1 second. You said von Humboldt. Did you mean von Helmholtz?
No, well, so Helmholtz and
was von Humboldt too?
Yeah. So Alexander von Humboldt in the 1790s, he's watching Galvani and Volta have this fight, and he starts looking into it as well, because he's also this sort of, he's got 5 brains tied behind his back. He's a big polymath, and so he starts experimenting. Although I keep saying like this, the polymath. I think science was the structure of science was different then. People could, at some point in the 1800s, it was still possible to know everything that was going on in science. Like today,
because we didn't know that much yet.
Exactly.
Yeah, exactly. And we hadn't, and everything was connected to everything else. I mean, it is now too, and we realize that. But but there's a time in the middle where we're like, yeah, I just got to work on this one very specific problem.
No, totally. Yeah. And like today, you can spend your entire career like working on like a single gene in a red-eyed fruit fly, right? And that's the sort of aspect of science that you're advancing. But like you maybe aren't, you don't care about what happened at the Large Hadron Collider,
Right.
But basically, like, yeah, so there was a point where it was possible to know everything in science. And so there were people who were able to, because of money and whatever, and brain power and, other, aspects of their lives, were able to actually be across all of this stuff, read all of the papers. When you read the papers at the time, they were so legible. You sometimes, you read a paper now and you're just like, oh God, my brain hurts after, 10 sentences, because obviously everything's become so specialized. People need to communicate in shorthand and jargon. But at the time, they were all communicating with everybody else. So like, a physicist would write something that, a person who was studying medicine could understand. Anyway, sorry, this is another rabbit hole. Jeez.
No, but it's good. This is what makes it rich. I love it.
I guess. So von Humboldt starts doing electrical experiments as well. He goes pretty nuts, actually. He does self-experimentation in probably the most shocking example in my book. I was so delighted when I found this. You can't even imagine. He was trying to understand how electricity affects biology. And instead of zapping a bunch of, sort of farm animals or, frogs or dead prisoners, he does it to himself. And so he threads, in one case, a wire into his rectum that's connected to a galvanic battery. And he starts to see sort of sparks, and he's got like terrible abdominal cramps and, the everything you can imagine that goes with the cramps. And instead of stopping, he just goes deeper, and he's got like visual, hallucinations. And, he went as far as possible always in all of his, I think, scientific endeavors to sort of understand what was going on. He had a friend, Ritter, who also took part in these experiments of electricity on the sort of natural electric variety. He also did these wild experiments, and I think he fried his nervous system so much that he died at 34.
Oh, wow. So he was also doing self-experimentation.
Yeah, they were doing.
So, I mean, it's cool. Some of the coolest stories. I feel like I want to read a book about people who did self-experimentation.
Oh, I'm sure
you see
there
examples
must
of it.
Be a good one. Yeah,
it's so cool. Like Newton did a lot of weird stuff to himself, like vaccine. A lot of people were like, "I'm trying to do this thing, and there's too much red tape. Well, I'll just inject myself." And it's like, whoa, this is amazing.
Well, yeah. I mean, the wildest example of that, I think, is the neuroscientist Phil Kennedy. He put his own brain implant into himself. He had a surgeon implant it. Yeah.
Oh, wow.
I don't know if I'm not sure it was, I have to be careful here with legal language. I'm not sure it is allowed to do such a thing. So he left the country and paid someone
international
to do it
waters.
For him. But it was not in the US, because I don't think it would have been. But because he was trying to get regulatory approval for an implant, but the FDA wasn't sort of cooperating. So he was like, look, I'll just get my own data to prove this. And I mean, yeah, the results were pretty wild. Anyway, so back to von Humboldt. So he basically, he is obviously plugged into this huge scientific network. So he recruits this guy, Johannes Müller. He's the hub in this big scientific network. And so he's able to connect people like Emile Dubois-Reymond, get them working at Müller's university in his lab. He's able to find people like that guy Matteucci I was talking about before, who had made the frog thigh battery. He takes that paper, he passes it to Müller. Müller gives it to Emile Dubois-Reymond, and then Emile Dubois-Reymond basically runs with it. And
so this is just like rich, smart people talking to each other.
Yeah.
Okay.
This is what it was at the time.
Von
That's
Humboldt,
what science was.
Right. That's what science was. When I think of von Humboldt, I think of him as like a naturalist.
Yes,
like an explorer.
Exactly. Yeah, but
not
I mean,
like somebody
he
who's
also
like tinkering in animal electricity, but I guess he was.
Well, he just had so many interests, that it was really surprising when I saw his name come up all the time. I was like,
that's why I was confused. I was like, Helmholtz?
Yeah.
They were like, no. I was like, I know that name from my classes.
Yeah, no, von Humboldt was involved in sort of making sure all of these people were connected up in the right way and working on this stuff together. So
that's cool.
Like networking, the LinkedIn of the day. Oh God, I'm making the most horrible comparisons.
So LinkedIn for rich white dude. Oh, it's the same.
Yeah.
It's the same.
Yeah.
Okay. So tell me more. So we know that these guys now are connecting with each other. We're starting to see a little bit more maybe legitimacy.
Oh yeah, super, super much. I mean, at this point, you've got them measuring the speed of the action potential.
Oh wow! Okay, so
your friend Helmholtz, he actually zeroes in on it, and he's like, the nervous impulse travels at the speed of a race car. So, which then again opens a huge can of worms, because people are just like, if it's electricity, how can it be going at the speed of a race car? Because I thought that just went really fast down a telegraph wire. So there's all this fight. There's all these fights about like whether it's electricity. Is it? Isn't it? What about chemistry? There's this whole war between the soups and the sparks. It's like science's own West Side Story.
But it's both.
Yeah, exactly. But it takes a long time to figure that out. And the culprits in this, but it's both, are ions. Yay! Ion channels.
Yeah, we got there.
Yeah, exactly.
I love it.
So this is something that we finally sort of put to bed only in the 1970s, when
Wow, really?
Yeah, Erwin Nair and Bert Sackman develop a tool called a patch patch clamp, which is my favorite.
I know it well.
Yeah. Oh, do you?
Just so you know, my master's was in neurobiology, and I worked in an electrophysiology lab.
Oh, well then, yeah.
So
I don't need to tell you this.
Yeah, we didn't but the audience probably did. We didn't patch clamp much because my professor, who's a German guy, he developed something called an MMEA or a MEP. It was a micro multi-electrode array that he designed himself, and we would build them in the lab, and you would plate neurons on top, and it could record 64 channels at once. And we had really good software that helped you discriminate the different action potentials that were coming from the different cells.
Oh,
and is it coming from an axon or cell body? Yeah, it was really interesting, so that we could look at network level electrical response as opposed to just single cell electrical response.
That's really cool. Yeah,
it was fascinating, and it was quite sophisticated and really interesting. But yes, I had to learn all about. This is why I know Helmholtz, because Dr. Gross would always say, "This is the Helmholtz double layer," and it has been burned into my brain. Yes, so there's that.
Well, right. And so, yeah, so I guess I'll do the sort of ion channels for dummies now.
Yes, please.
Because this is the only way I understand it. But great. So you'll know more about this than I do, but I will.
Or I will have forgotten more than I have ever known. So this will be helpful for me as well.
So keep me honest. So just make sure that I've understood this correctly after bleeding from the eyes for several years, trying to learn membrane biophysics. But yeah, so this is the ion channels for dummies. So your cell, all the cells in your body, neurons, any cell, is basically its insides are separated from its outsides by a membrane. We won't talk about lipid or bilayer lipids, but it's like
lipid bilayers.
Let's call it a shrink wrap, because that's how I like to think of it. So it's a shrink wrap, but it's got loads of little holes in it. It's perforated by tens of thousands of little holes. These are the ion channels, but these are not plain old holes like in a sieve. These are smart pores that can decide what to let in and what to let out. And the stuff that's outside your cells is similar to the consistency. It's got similar constituents as seawater, right? You've got sodium and potassium, calcium, chloride, things like that. But basically, your cells, they like potassium, they don't like sodium, and so they let in preferentially potassium. Sorry, this is neurons in particular. They let in potassium, they keep out sodium. And the sort of pressure, the electrical pressure that this exerts on the cell to separate the ions this way, because ions, sodium and potassium, they carry their own tiny electric charges with them. So when you separate them, you create a condition where the inside of the neuron is always at a slightly lower electrical charge. So it's about a difference of minus 70 millivolts inside the cell compared to what's outside the cell. Sorry, I'll take a breath.
So good. It's so good. And it's always kind of hanging at that level until.
Yes, because that's its resting potential. So it's kind of like its happy place. That's where the neuron likes to put its feet up, have a cup of tea. But when the action potential roars through, it's basically Panic at the Disco, and all the gates open, and sodium swaps with potassium, like dogs and cats living together, and then everything swings wildly. The voltage changes violently. For a minute, it all goes positive, and then it goes back to zero. And then all the ion channels are trying to just get everything back to normal, kick out the sodium, bring back, the potassium where it belongs. And that is how that's how the action potential moves down a nerve cell to the next nerve cell. Of course, there's a synapse involved, and that's the part where it's also chemistry. But without the sort of electrical signal, you wouldn't have it. Your synapse could, all the serotonin in the world wouldn't pass your signal along. So
right, and there's a reason that we literally say the neuron is firing, fired, because that massive depolarization and the repolarization, like you said, it's Panic at the Disco. It's fast, it's violent, the neuron is it's firing to the point where when we transduce the signal, you can hear it you can hear it on a
Oh
speaker.
yeah. Oh
Yeah.
yeah. Sorry, I was reading that in Ashcroft's book. Frances Ashcroft wrote this beautiful book where she's, it goes into some of the history of like the voltage clamp and like how they used to be able to just hear the pops.
That's what we
So
would do. We would
oh
actually set our MEAs up to a speaker, and you would listen to your neurons chattering the whole time. And that's how you could tell when you would add a drug and their chatter pattern would change. Because our minds are organized. I mean, we're organized visually, auditorily, everything, but we're musical species, right? So we can hear patterns. And then when those
oh,
patterns
that's
change, we can
so
go, oh, the
cool.
drug's working now. But
Oh my
yeah,
gosh.
like watching it is sometimes not as easy because you tend to look over here and over there and you're seeing all this stuff, but you can hear the music of the cells
Oh, that's so
easily.
cool. Oh, wow. I wish I had interviewed you for the book, to be honest.
I know, but it would have had to have been like
Oh,
15 years ago when I was still in the lab. Like I'm
well,
pretty
that's super
rusty on
interesting.
this.
So these were ion channel like blockers or openers, I assume.
Yeah,
Sorry, now we're
depending on
getting
what we're
super
using.
nerdy.
No, it's okay. I love it. And I think deal with it, people listening. No, there were different drugs we would use. So there are things called like bicuculline. Like there are drugs that actually induce kind of like kindling. So basically the neurons tend to in a cell culture. So this is outside of the brain. So I've basically taken individual cells from like embryonic mice, and then I've taken them out of the mice, and then I've plated them, and then they've regrown into nerve networks on their own. Like they naturally organize. They tend to fire in a predictable pattern where they all fire together. Bzz, bzz, bzz. And you can use different drugs to induce that pattern more or to make it a little bit more chaotic. And so what we would often do is induce a predictable pattern, and then we would do changes, whether it's pharmacologically induced changes or oxygen or water or trauma, whatever the case may be. And then you could actually hear the patterns change. And so, I mean, we had different researchers doing different things with them. Some were studying Parkinson's, a model of Parkinson's, some were studying other things, but also some were trying to develop biosensors. So basically you put this somewhere and then you add a new chemical, and then what happens to the
Oh,
cells?
totally. Yeah. Oh, yeah.
Yeah,
That I'm
yeah.
obsessed with that because, of course, like the whole biocomputing,
Right, right,
bioprocessing thing is, I feel like just like criminally understudied. I feel like
right.
more resources should really be thrown at that stuff.
Oh, and we got no money ever. And
Yeah.
I think one of the things that was really fascinating about what we did in the lab, which I think you do see this in other labs, is you can do a lot of basic science. You can literally just look at the cells and go, when that cell fires, that one does, but that one doesn't. When those 3 fire, then these 7 fire afterward. There's so much computational neuroscience that can be done
Yes.
when you have a network of cells just doing what they do, because you can map them.
Yeah.
Yeah. So
Oh,
there's a lot of just that. We had physicists in our lab too. My role was wet lab. It was boring. Just so in case anybody's interested, my master's project didn't really work because I was trying to do neurotrauma and the machine kept breaking our MEAs. So
it was a
ultimately
different
at the
kind of
last
trauma.
minute,
Just
it was
for
a different,
you.
totally. Yeah. So ultimately me at the last minute, I actually wrote a full protocol for running the wet side of an electrophys lab because I was the cell culture, like the chief cell culture technician. So I have a protocol soup to nuts from the mouse all the way through all the technology, through all of the chemicals that you use to plate these things so that you can use them for research.
Well, so that's really cool because I really wanted to get into ion channel drugs later in the conversation.
Nice.
Because
Yes.
Well, because so we figure out this whole thing with the bioelectricity and like the fact that it's real, it's not an epiphenomenon. It's not just like the exhaust coming from the car that tells you it's working. It's actually like a functional thing. And I guess like once we figure out ion channels, like basically immediately they start trying to or working on drugs that either, prop some of them open or keep some of them closed. And there's this huge explosion of ion channel drugs. And that's really interesting. But like people know about the patch clamp and they know about ion channel. Well, I don't know if they know about ion channel drugs, but there are some tools that nobody ever talks about. Like my favorite, the vibrating probe. Like, were you familiar with the vibrating
No, we
probe
did
at
not
all?
use a vibrating probe at all. Tell me more.
Sorry, this is not what you think it is,
Okay.
given the context of
yeah, right, of the previous conversation.
Sorry. So no, well, it's just that like within the context of neuroscience, bioelectricity research was going from strength to strength, but there were still aspects of bioelectricity that had nothing to do with neurons or brains. And people just like came across these things and they were like, oh, okay. And nope. So for example, galvanotaxis, I'm sure you must have seen this a bunch of times. Like if you're, so you take a cell, like any cell, a plant cell, like oat seedling, whatever. It's all of these cells, when you measured them, anything like algae, oat seedlings, some random seed of some stuff, skin cells, anything, all of these things, all these living things, anytime you measured it, you found a voltage. Like,
right. They all have a resting potential, right?
yes.
We only consider it, quote, excitable tissue if the difference between the inside and outside is really, really big.
Yeah. And if it changes, but there were some.
And if it changes, yes. So
yeah.
that's like muscle, heart, nerve. What else? What other excitable tissue do we have?
Is glia, does that count? But I mean,
yeah, probably. Yeah, also, yeah. So
but that's basically it.
Yeah.
Like, what about bone cells, skin cells? What about like blood cells? What about, plant cells,
Right.
So, actually, just tiny aside, Nightella, which is an algae of some sort, I think, or some sort of water plant. Nightella was a major locus of electrophysiology research before we got the voltage clamp. I found this out writing a chapter that did not make it into the book because my editor was like, "Oh my God, please stop writing more." He was like, "Please stop. Can we just keep it to the body?" But I was like, "What about plants? Plants are electric. Fungus, it's electric." Anyway, but I went into a super deep dive on plant electricity as well because that is super interesting. Nightella was like the main locus of electrophysiology, like membrane biophysics research but that's completely beside the point. I promise I'm going to stay on topic. So the first time we're able to actually take the real measurements of these tiny non-action potential voltages is in the 1970s because this another sort of obsessive botanist, Lionel Jaffe. I mean, he was all kinds of scientist. I mean, I'm calling him a botanist because he loved marine fucus, which is algae, brown algae, because it's a sexual creature and it has sperm and eggs, but you can watch it develop without, having to deal with a womb or, deal with, animal protocols. You can just basically watch like the egg develop after having been, fertilized.
Gotta love a good model organism.
Yeah, exactly. Yeah, exactly. So he's just like, I think this thing is electric because it grows in the direction of sunlight. So on the one side, it develops its little holdfast to grip onto something. And on the other side, it grows the frond to, grow toward the light and photosynthesize. So he starts, and he's just like, okay, so how am I going to measure whether this thing has a voltage? So he gets his grad student, Richard Nuzzatelli, to make him a device, and somehow everybody keeps a straight face while calling it the vibrating probe. I mean, there's a super boring, long, tedious explanation of why that's exactly the perfect name for it. And, it probes. It also vibrates,
Right.
But, anyway, so he develops this vibrating probe, and this thing is the most sensitive thing that had ever been developed. It was able to read these incredibly faint fields. And with it, they start measuring everything. And everything has this very faint voltage. And they call it the physiological field. And then they take this physiological field, they make a fake version, obviously, just like whatever, they just generate this electric field. And they apply this electric field to any number of cells in a petri dish. And every single time, the cells start migrating, like physically crawling across the petri dish toward the cathode end. but like they had known this since like the 1920s, but scientists saw this and they were just like, number one, that's gross. I mean, I'm sure they didn't, but like that's what I would have done. But they were just like, look, this is illegible in biology. Like, screw this. I'm not like investigating it any further. Let's politely ignore it and hope it goes away.
Right.
And so Jaffe is the first group. Jaffe's group is the first to start being like, well, what the hell's going on? And it takes like 40 years. 40 years later, Nuccitelli, the guy who invents the vibrating probe, he finally invents a device in 2011, I think, called the dermacorder that's able to measure the electric field that you emit when you have been wounded. It's called the wound current. Dubois-Raymond was the first one to actually notice this. He had no idea what it was. It kept getting in the way of his measurements. He was super annoyed by it. But again, he was like, "No, what is this? Fuck this! I'm gonna, I'm after the action potential. Like, stop distracting me." 2011, Nuccitelli finally finds a way to measure this thing. So what happens is skin cells, right? They're just like us. No, sorry. Skin cells are just
they
like
pump their own gas. Oh, sorry.
Sorry, it is late and I'm starting to get delirious.
Love it. It's
Sorry.
the best.
So skin cells also have all the ion channels. They also are similar to neurons in having a resting potential. Well, not resting potential. But they have an inside. They're minus 70 millivolts inside.
Well, they also come from the same developmental tissue. Yeah,
yes.
They're neural tube. Yeah.
Yes, exactly.
Yeah.
So those guys have been friends forever. So
right, yeah.
But what happens is in your epithelium they are polarized, so they all line up, like they kind of link arms, and they sort of they all line up in the same direction. Basically, they're all facing out. Basically, with their and what this does, it creates something called a transepithelial potential, which is just like this steady sort of field. You cut this, right? You get a cut, and that short circuits this current, and you end up with basically just current flowing everywhere. And this creates an electric field that is measurable. Now, this is going to blow your mind. Okay. The whole point of the electric field is that it calls cells like fibroblasts and macrophages. It basically makes those guys crawl in, and they all go toward the center of the wound. Now, guess what that is? That's the cathode of the electric field. It's like a big fucking bullseye for them to be like,
wow, that's so cool. So the last thing you want to do when you cut your hand is like send a bunch of current through it and confuse all of your cells. You want to let them naturally do what they're doing.
So, yes, broadly. So the thing is, people have, ever since Nuccitelli sort of showed this, sorry, and I need to add one more thing. The wound current, it spikes basically when you first got injured. And as your body repairs the wound current gets sort of weaker and weaker and weaker until when it's completely healed, there's no more wound current. So that's cool, right? But then also, the wound current is twice as strong in people under 25 as it is in people over 65, which is a super interesting correlation, I think, with the fact that you heal more slowly as you get older. So it's really interesting. I wish more research were done on this, but like, it's just the thing that's frustrating is that like the only place that bioelectricity still is really legible is in neuroscience, because people are like, oh, of course, like action potentials and like brain currents and like, body electricity, electrical nerve signals. But they're just like, but I'm just like, everything is electric. Like, and
and I guess maybe in the heart, like people, get the heart nodes too. But you're right. Like, so this is the thing that's making me very sad right now, Sally, which is that like, we're coming towards the end of the hour. I have another, I know, I have another podcast right after, and we haven't even talked about like the future or like the cool, weird stuff that like, so maybe we can take,
I'm such a rabbit hole person.
Stop. I love it. It's amazing. I want to have you back on. I want to talk to you more, but maybe we can whet, I hate that word, people's appetites a little bit with like one or two things that as you were digging in are like the most interesting, the most fascinating, or the most exciting for you about sort of how what we know or what we hope to know soon could be applied to future therapeutics or interfaces or
Oh my
means for
gosh.
understanding the world more.
Yeah, no. Okay. So the thing that you were saying, like, should you not apply an electric current to a
Right,
wound?
right.
Well, we don't know entirely, but it looks like if you apply an electric, if you know the right parameters,
Right.
it is
Yeah.
possible. It is possible to accelerate healing, and there is a huge DARPA program that is actually looking to do exactly that. And it's not
No
just with electric
way.
current, but they're using ion channel drugs to try to keep to try to figure out like what is, the electric state of a wound and how does it interface with the other type, sort of the chemical state of the wound and, all the other like the anatomical sort of healing stages. The goal is to cut the healing time in half by affecting the bioelectric sort of properties of the wound. And a lot of researchers are looking at all of this. I didn't even get into like regeneration, the fact that like some animals are able to regenerate whole limbs, where for us, like after age 11, it's game over for us. Like, and before age 11, it's also kind of game over. Like all we can really do is regenerate the tip of a fingertip if we lose it. But
And even if we cut through anything central nervous, we're kind of screwed too.
yeah,
Like that
totally
stuff's not
screwed.
going to get better. Yeah, yeah,
Yeah.
yeah.
So there's a lot of, so I think the sort of probably the most well-known bioelectricity researcher right now is Michael Levin, and his thing is that he is looking for the bioelectric correlates of regeneration. Can we like what is it that we're trying to do? Are we trying, because the electrical profile of a wound in an animal that regenerates and one that doesn't is very different. It's
Okay.
a really interesting
So there's
voltage
something in that.
difference. Yes, and he's
Yeah.
sort of after that, and he really wants to. There's a lot of people who are looking at how, like, whether we can regrow entire limbs in frogs and mice. And, there's all these like ion channel cocktails that people are investigating. Some people are actually just using actual pulses of electricity. It's super early days, but they have gotten way further than anybody has before in growing these usable, not perfect limbs, but like way better than you would imagine that a non-regenerating animal can grow. And then there's cancer, taking it back to, the sort of ridiculous, as soon as you ever say the word cancer, people are like, yeah, sure, of course. Cause like, whatever cleanse is meant to, cure cancer, then there's, like there's just every quack knows how to cure cancer. Right.
But
But
also cancer and aging, cancer and cell repair. I mean, this is at the heart of
exactly.
it. Yeah.
Levin always said to me, he said that like at the end of the day, all of these problems, wound healing, regeneration, development, cancer, they're all a matter of like what shape, like a collection of agents, your cells, choose to assemble themselves into, and how to make them do one thing when they would rather do another. And it's all the same thing. And I mean, we didn't even get into development. I'm sorry, I'm just filled with regret with all the sort of asides. Like who
Don't
needed to know
be.
about Alexander von Humboldt's rectum?
But here's the thing. Here's the thing. All the stuff is in the book, and we don't want to read the book to them out loud, right? We want to get them excited about the book so they read it themselves.
I know,
and I think we did that successfully.
Do you? Because I'm just like, oh, there's like all this cool stuff with ion channel blockers and cancer. Oh, all right.
Well, but maybe you could come back and maybe we could do a part 2, because we were talking about what was, and now maybe we can talk about what could be. What do you think?
I would absolutely love that. I would love that
Me
so much.
Me too. And usually I wouldn't fly through and I wouldn't be like trying to wrap it up. I would just go for 2 hours. But I'm headed to Jordan in 2 days for this like intense trip to the Middle East.
Oh, wow.
And my assistant has been like really on me. She's like, Carrie, you need to record more episodes. You don't have enough episodes for when you're gone. So that's what we're doing now. But you have been like an absolute, I mean, this has been like one of the most fun conversations that I've had in a really long time on the show. You're just such a pleasure. I do want to talk again. Can we do that? Can we make a plan for that?
I would absolutely love that. Like I'm a little bit, like lots of stuff is happening right now. It's my first book, so I just don't know what happens I don't understand any of it, but but yeah,
maybe after because it's about to launch, or it's about to land.
yeah.
So, and of course, everybody listening, it's either out now already, or you can definitely pre-order it. So make sure you're going online to buy a copy of We Are Electric: Inside the 200-Year Hunt for Our Body's Bioelectric Code and What the Future Holds. And that is by Sally. I'm going to try and say it right this time so I don't screw it up later. Sally Adee.
Yes, it's perfect.
Yay! I did it. Thank you so much.
Can I just say, I really appreciate our conversation. I get really nervous before interviews, and all I wanted to do was just chat to you, just do Aldini chat.
Oh, good.
And I had such a good time.
I'm so glad. That's all I ever want to do anyway. Oh, me too. I feel like I do this so much and I've been doing it for so long that I don't think I could do it if it wasn't fun. You know what I mean? I wouldn't have the motivation. And so I'm so glad that you had fun, because I know I did.
You made me so unnervous that I forgot to say every single thing that I meant to say.
But you said all the good stuff that you get. People can listen to other interviews for that stuff. This is our interview, damn it.
No, that's true. I didn't get into any of that fun stuff in any of my other interviews. So thank you.
Right.
Thank you. It was so nice to talk to you.
Oh, thank you so much. Yeah. And hey, everybody listening to the show, thank you for coming back week after week. I'm really looking forward to the next time we all get together to talk nerdy.